Hello students, in the last session we have completed tubular reabsorption and tubular secretion in DCT. Okay. Now last part is remaining of mechanism of urine formation, okay, that is tubular reabsorption and tubular secretion in collecting duct. Okay. So let's study which are the substances which will be reabsorbed and which are the substances which will be secreted in the collecting duct of nephron. Okay. So this is the collecting duct of nephron which receives almost 8 uriniferous tubules. Okay. Likewise, many such collecting ducts will be present in the kidney. Okay. So this is one collecting duct which I have drawn, and whatever these outlets which are meeting here in the collecting duct, these are the openings of the uriniferous tubules. Okay. So 1, 2, 3, 4, 5, 6, 7, 8. So 8 uriniferous tubules are opening here in the collecting duct. ओके सो ये यूरिनिफेरस ट्यूब्यूल का DCT जो है वो DCT नेफ्रिक फिल्ट्रेट यहाँ पे छोड़ता है कलेक्टिंग डक्ट में. सो नेफ्रिक फिल्ट्रेट विच इज़ कमिंग इन द कलेक्टिंग डक्ट, okay, which will have very less amount of important nutrients, okay, because बहुत ज़्यादा रीअब्जॉर्प्शन तो PCT में हो चुका है और DCT 90 to 95% सोल्यूट्स जो बचे हैं नेफ्रिक फिल्ट्रेट में वो भी रीअब्जॉर्ब कर लेता है. ओके, गॉट इट. सो here in the collecting duct majorly there will be reabsorption of the water. Okay, so large amount of water will be reabsorbed from the nephric filtrate of the collecting duct because water हमेशा लगता ही रहता है body को. So it will try to reabsorb maximum amount of water, and to some extent urea is also reabsorbed. Okay, so this urea will enter in the interstitium. Okay, so interstitium में urea reabsorb होगा and from the interstitium this urea will enter in the blood of peritubular capillaries. Okay, so why this urea should enter in the blood of peritubular capillaries? To maintain the osmolarity of the blood. Okay, so isn't it collecting tubule is also involved in maintaining the osmolarity of the blood as well as it also maintains the ionic balance. How it will maintain the ionic balance? अगर blood का pH कम हो गया नहीं तो ज़्यादा हो गया, इसका मतलब there is either less amount of H positive ions or more amount of H positive ions respectively. Okay, so H positive ions अगर blood में ज़्यादा हो गए तो there will be removal of the H positive ions from the peritubular capillaries in the collecting duct. Similar way, K positive ions, if they are more in body fluid, then these K positive ions will enter in the interstitium and from interstitium they will enter in the collecting duct. Okay, got it. Thus collecting duct will maintain H positive ions and K positive ions in the bloodstream. Okay, so collecting duct is involved in maintaining pH of the blood as well as it is involved in maintaining ionic balance and osmolarity also. Ionic balance by releasing H positive ions and K positive ions, pH by releasing H positive ions, and osmolarity by reabsorbing urea. Okay, got it. So here I have drawn the diagram of collecting duct wherein collecting duct extends from renal cortex till inner side of the medulla. Okay, so this is the region of the collecting duct in the renal cortex, this is the region of the collecting duct in the outer medulla, and inner medulla में भी extend होता है collecting duct, and many such collecting ducts will join to form collecting tubule, and collecting tubule will then open in the duct of Bellini. Okay, got it. So this is all about tubular reabsorption and tubular secretion. सो ट्यूबुलर रीअब्जॉर्प्शन किसका होता है? कलेक्टिंग डक्ट में large amount of water and small quantity of urea to maintain osmolarity of blood, and tubular secretion of H positive ions and K positive ions. If H positive becomes more, to maintain proper pH of the blood, H positive will be secreted out in the tubule of the collecting duct, and if there is more amount of K positive ions in the blood, these will be secreted out in the collecting duct, thus maintaining ionic balance in the blood. Okay. So here we have completed mechanism of urine formation. So mechanism of urine formation में बहुत सारे video lectures हो गए अपने. Okay, we have started with ultrafiltration. Okay, then after that I have explained you difference between tubular reabsorption and tubular secretion. Then we have completed tubular reabsorption in PCT, then tubular secretion in PCT, followed by tubular reabsorption and tubular secretion in we have then completed the loop of Henle's part, what is actually reabsorbed from loop of Henle's, and then we have completed tubular reabsorption, tubular secretion in DCT, followed by tubular reabsorption and tubular secretion in collecting duct. Okay. So this is all about mechanism of urine formation. Thank you.